Okay, thank you for joining us today at the 2022 Spring GES Symposium. Just a note that we are recording the session today, so if you prefer not to be recorded, then please just turn your cameras off and leave them off. So as part of a GES, the GES degree requirement, every GES student is required to conduct faculty mentored research experience in which they write a thesis and present their findings along with taking questions to a public audience. So that's what we're here to do today. So we have 15 presenters today, which is a record for the GES program by about 50%. So in 2018, we had 10 presenters. Accordingly, today's proceedings are going to be broken up into 2 sessions. There's this one this morning, which will have 8 presenters, and then there'll be another one this afternoon with 7 presenters. And that one this afternoon will start at 1:30 p.m. Because of the large number of presenters, we need to keep to a tight time schedule today. So 15 minutes have been allocated for each student, during which they will be briefly introduced by their faculty mentor. They will give their presentation, and then they will answer questions from the audience. After the completion of each presentation, you can briefly unmute and congratulate the presenter. Otherwise, we ask that you keep your microphones muted and hold putting any comments in the chat until the presenter has completed their presentation. I will be moderating the Q&A portion. If you have a question, please use the raised hand under reactions in the Zoom window, or just type your question into the chat, or type into the chat that you have a question. Because of the tight times table today, I apologize up front if we don't get to all of your questions. If you have questions that you didn't get to ask, please do the private chat to Latina, and she will keep a list of these extra questions, and we'll ask the presenters to stay on at the conclusion of the symposium, and they will address some of those questions at that point. Okay, without further ado, I'm going to just introduce the faculty mentors, and they're going to introduce the student presenter. So without further ado, our first presenter will be introduced by Dr. Kristi West from the Hawaii Institute of Marine Biology.
Thank you. Sara Bauer grew up in California, but has made the most of opportunities associated with her time here in Hawaii, illustrated by this photo taken at our lab with Sara at the far right. Sara has been a consistent presence in our lab over the past year, bringing positive energy, a high level of curiosity, and an interest in continuing to learn and explore new things. Today, Sara will talk about her thesis research, and with that, I will hand it over to Sara.
Thank you for introducing me, Kristi. So the research that I did is determining the rate of DNA degradation in pygmy killer whales for the estimation of a post-mortem time interval. So to start, why do we care? Well, pygmy killer whales in particular are a rare species, but moreover, they are marine mammals and they're vital to ocean ecosystems. And we're looking at pygmy killer whales in my research really to set a precedent so we can use these findings for further research. So why do we care about the post-mortem time interval? Well, the post-mortem time interval tells us how long these animals have been dead. So essentially when we find them on shore, how long have they actually been dead for? When did they die? And understanding this time of death is really important for us when we're necropsying the animals and we're trying to determine what might have caused the death of this animal. So now, introduction to DNA degradation. Well, the DNA degradation has been proved in forensic science to be used as a tool that we can use to determine post-mortem time interval. So DNA is a relatively stable molecule, and once you or any animal dies, your tissues break down, and the DNA also breaks down just like that. So what we were hoping to find was something similar to forensic research, where we find a linear rate of decay of DNA concentration. And we found this, or previous research has found this in rats, pigs, and humans, but it's never been done before in wildlife. So what we were hoping to see is some kind of negative trend line, like in this image, where you start with a high DNA concentration at day 0. That would be around the same DNA concentration you have when you're alive or the animal is alive. And over consecutive days since death, this DNA concentration would go down. Thus, it can be used as a tool to kind of determine when something died, if we can only look at the DNA concentration. So how did we do it? Well, we used two freshly dead pygmy killer whales. They were from a 2019 stranding in Maui, you might have heard about. There were 11 stranded pygmy killer whales. And we call them freshly dead because they were euthanized. We put them on ice and they were brought by air cargo, necropsied at our Hawaii stranding lab, and then they were put in a neg 80 freezer. So that was to prevent as much DNA degradation or just degradation of tissues as possible. So we used 7 different tissue types of these animals, and then we put them into 2 replicated stranding environments. And that's because when we have stranded marine mammals, we either find them on shore, on the land, or we find them in a water environment. So what we did is we took all of these tissue samples and we put them in a replicated water environment. So if you see in this left-hand picture, we put them in these boxes under the water that we could just pull out. And the replicated air stranding was just left on the pier. And it was just in these opaque containers to prevent any UV degradation or introduce other variables. And we sampled the tissues on 2 different timelines, essentially. We tried to look at a 7-day time interval to see if it might be more precise, trying to determine a trend line via that versus looking at 28 days, because previous research has found shorter time intervals to be more reliable. And with those 7 different tissues and with those several intervals of days to sample on and 3 replicates and 2 animals, we had 714 samples. So next, what did we do with all of these samples? Well, after they degraded, they were put in a neg 80 freezer to, again, prevent any more degradation. Then we subsampled 10 milligrams of tissue from each of them, which included things like liver, lung, air, lymph node, blubber, muscle, and rib marrow. So we took those 10 milligrams, we added lysing agents and incubated them in a hot water bath to break everything down, kind of homogenize everything, and to make it so that the DNA was something we could extract. Then we extracted that from this solution, and we used then a qubit fluorometer to determine the DNA concentration that was in that 10 milligrams of tissue. And the way we did that, you add a binding agent to the DNA. So all the nice, pretty double-stranded pieces of DNA, but it would bind to, not anything that was broken down. And then we shot kind of light on it using the fluorometer, and everything that had binded to this agent would kind of bounce back light. So the results, well, if you can see, we have this nice graph here, and it shows the averaged DNA concentrations over a 7-day interval. So this is averaged between both individual and between the stranding environment. And that was because when we did our statistical analyses, we found a negligible difference between individuals, which wasn't surprising. Previous research has shown there's little to no difference between individuals. But what was more surprising was there was no difference between the water and sea environment. So we were able to just average everything. So looking at this, we can see that there's 3 lines with higher slopes. So starting with the light blue, that's the lymph node, the light green, that is the lung, and the red is the liver. Then down below, we also have muscle, cerebrum, and blubber, and you can't really see the difference between those. They have kind of a flat slope line. And this was across the shorter 7-day time interval. And if we look at the 28-day, we have something that's pretty similar. So it's those 3 same tissues again that have the increased slope lines, the lymph node, liver, and lung. So looking at this, we did create the graph that I showed at the beginning. We had these negative trend lines. Essentially, we had a high starting DNA concentration, and over time, we did find that this DNA concentration went down. And it went down pretty linearly as well, at least for my 3 favorite tissues now. So what we did next was to look at the R² values, and these R² values help tell us how significant the relationship was between the DNA degradation concentration going down over time. And we found that those 3 tissues that had the increased slopes were also the ones that had the best R² values. So you have the liver, lymph, and lung highlighted on the left there, and you can see the R² averages in the 7-day on average are much higher than the 28-day, 0.5 versus 0.3. And then especially on the 7-day, liver, lymph, and lung had much stronger relationships. So next, to kind of summarize those results, essentially we had significant relationship between the PMI and the tissue type. Each tissue had a very distinct starting concentration. And they all had their distinct slope lines. Then we also found no significant difference between our individuals, between the 2 pygmy killer whales that we used. And then there was no significant difference between the air and water stranding. We also found that the shorter PMI was much more reliable, as you saw with the R² values being much higher for the first 7 days. And then we found that the liver and lung were the most reliable tissues. And a note on that is, while lymph node was part of those 3 with the really nice high R² values, we found that the lymph node had a higher coefficient of variability. Essentially, there was a lot more difference, or higher variation between our 3 replicates that we did. So we didn't find them to be, or the lymph node to be as reliable as the liver and lung was. So some discussion on that is that to start with the air and water difference, it's not that surprising here in Hawaii that there is little difference between those 2 conditions. And that's because we put temperature probes in, and there was not a huge difference in the average temperature. And we are, in Hawaii, where it's both warm in the water and in the air. But if we were maybe at a higher latitude, we might expect a higher difference, just because the water would be much colder, and thus it might help prevent the DNA to degrade at such a high rate. However, in Hawaii, that research, or any research using this tool, you probably won't find a large difference between a water and sea stranding. Next was that if we are to use this research in the future, it's going to be the best if we use the maximum amount of tissues as possible and try to line up the concentrations of each tissue with that graph, and then to use it in tandem with the observational methods that we already use. But again, the observational methods are not going to be as reliable as something quantitative, which we don't have yet. Next was something, or a couple things for future research. I would really want to look at homogenizing a larger sample of tissue. When we have the 10 milligrams and we lyse it, essentially we're homogenizing those 10 milligrams. I think if we homogenized a greater area, like just a huge chunk of tissue and homogenized it essentially using something like a blender, then we might have less variation between samples. And this is because we can find bronchioles and lungs and collagen that can have variations in DNA concentration. Future research would also look at variations between different species. So we might have a lot more difference between different individual species than individuals within a species. But yeah, that is it. So I'd just like to extend a thank you to my mentor, Dr. Kristi West, PH student Cody Clifton, and my lab mate, Alex Mathers, as well as the entire health and stranding lab that were just a huge help with getting this project done. I would also like to thank the Naval Sea Systems Command, as well as the UROP program for helping to fund some of this research. So big thank you to them again. But that's it. That'll conclude my presentation. Does anyone have any questions?
Okay, we can unmute and applause if you want. Okay, any questions for Sara?
I've got one. Nice talk, Sara. I was curious in looking at the graphs whether you had tried some nonlinear regression through those data points. It looked like some of the slope lines were actually changing over time, and maybe something like a negative exponential or something would have fit the data.
We haven't, no, we haven't done that yet, but that's a good point. Maybe we'll look at that in the future. Especially the lymph node, and that's why we didn't use it we didn't include it in the two main significant ones. It had that really high variation. So I hope, but we'll check it out. But hopefully with maybe homogenization techniques, it would also help prevent some of any variability. Thank you for the question.
We would have time for another question if there is one. Okay, great job, Sara. Okay, our next presenter is mentored by Dr. Sabine, who is currently in Europe. So she will be introduced by a graduate student in his lab, Noah Howins.
Thanks, Glenn. So Samantha first reached out to me in the fall of 2019 to seek advice for finding a research project. And when I met with her, I was seriously impressed at her level of background knowledge for someone who had just completed her freshman year in GES. She went on to officially join our lab in the spring 2020 semester and has gone on to be awarded funding and complete 2 Europe research projects. Outside of the lab and classroom, Samantha is an avid UH scientific diver and has recently become a certified dive master. Samantha has also been employed as a valued employee in the UH dive office for about 3 years. And this summer, she will be a dive staff member for the Catalina Island Marine Science Institute camp, supervising young divers in marine sciences. During the fall semester, she will be returning to Oʻahu to continue working at the UH dive office and potentially work with the Waikiki Aquarium. Good luck, Samantha.
Thank you, Noah, for that introduction. So again, my name is Samantha Darin, and my mentor is Dr. Sabine, and my thesis is on the determination of microplastics in surface waters of Kāneʻohe Bay, Oʻahu. So just a little bit of background on microplastics. They're defined as particles that are within 5 millimeters in length in any direction. So they're relatively small, and they can be broken down into 2 categories. The first category is primary microplastics. So these are plastics that were released directly into the environment as small particles. This can include abrasive scrubbers used in personal care products. So essentially, a lot of personal care products contain microbeads that are used for exfoliation purposes. And another example is plastic pellets used in industrial manufacturing. If you look at the image on the right, the top 2 boxes are microbeads used in personal care products. The second category is secondary microplastics. So these originate from the degradation of larger plastics. The image on the left on the bottom is the degradation of plastic bottles, and on the right is synthetic textile fibers from clothing. It's estimated that between 4 and 12 million tons of plastic waste enter the ocean each year. And based on this, plastic debris is expected to outweigh fish biomass by the year 2050. So this is a very significant issue. And it's also important to note that plastics don't ever break down completely. They just break down into smaller and smaller pieces remaining in the environment. Harmful additives in plastics can leach into the environment and can be especially destructive when mixed with other leached chemicals in the ocean. Common polymers in marine surface waters include polyethylene, which makes up plastic bottles, grocery bags, caps, and things like that, as well as polypropylene, which makes up most of the plastic packaging and containers that we see. So these are widely used plastics. And the reason why they're most common in surface waters is due to their low density. They tend to float. A study led by Caleb Brignac in 2019 found that within the main Hawaiian Islands, surface waters were typically dominated by polypropylene and polyethylene. So that is what I'm expecting to see in this research. My objectives of my thesis were first to determine effective sample collection and data processing techniques. This was a pretty big one because the field of microplastics is still relatively new, and there's not yet a lot of standardization on sample collection as well as processing techniques. So I did have to take some time to kind of dig through the literature and figure out what would work best for this particular project. My second objective was to gather data of microplastic concentrations using plankton net tows, then to digest that organic material and quantify plastics in the remaining particulates using Raman spectroscopy and FTIR. And then finally, to determine microplastic concentrations in the surface waters of Kāneʻohe Bay. So my hypothesis was that microplastics would be present in surface waters, which I defined as 0 to 1 meters deep of the sampan channel. I did expect, based on previous research, such as Caleb Brignac's study, that I would find mostly polypropylene and polyethylene. This is an overview of the site that I sampled at. So I sampled in the sampan channel, which you can see is depicted by the star there. And just for reference, on the left at the top, you can see the Kāneʻohe Bay sandbar. So the topography gets a lot deeper within that sampan channel. And you can see that most of the water in that area of the bay goes in and out through that channel. So I thought it would be a good place to survey. Just for more reference, on the right, you have the Marine Corps base with the blue marker. On the bottom in green, you have Moku'ulo'e or Coconut Island. And then on the left in pink, you have He'ea State Park. These yellow push pins show the 2 tow routes that I took when I was collecting data. So as you can see, there is one that is a little bit closer in on the inside of the channel and one toward the outside on the outer part of the bay. And I did this to account for any variability between those 2 areas of water. To collect my data, I conducted 10-minute plankton tows with a boat speed of 1 knot on the surface with a 53 micron plankton net. And that came out to about 0.2 mile tows for each round. I took into account the tow distance as well as the area of the net to determine the overall volume of water sampled, given that volume is equal to distance times area. I then collected all of the net contents in glass sample jars and brought them back to the lab. I was very careful to use glass anywhere possible as opposed to plastic within my sample collection as well as within my processing to avoid any contamination with external plastics. To process my samples, I utilized the NOAA marine debris method. The authors of this method stated that it was applicable for the determination of many common plastics, including polyethylene and polypropylene, the 2 that I was expecting to see more of. These solids were subjected to a wet peroxide oxidation in the presence of an iron catalyst in order to digest all of the organic matter, such as plankton. The solid material was then isolated into 200 micron, 500 micron, and 1 millimeter sieves to create size classifications. The remaining material was then mounted on microscope slides for observation, as well as determination of the composition of those polymers. The image on the left is a depiction of what a wet sample looks like. So before processing, this is after we've pulled the net out of the water. We use seawater to rinse all of the contents of the net, make sure it all made it into that glass jar. And then on the right, you can see a sample that has been processed and is ready for further analysis. So it's mounted on a microscope slide, and this example is tow number 7 with the 500 micron size classification. To determine the plastics, I first visually identified them based on morphology. So this is based on what plastics tend to look like under a microscope. You tend to look for rough and jagged edges or really flat, smooth surfaces and bright colors. These were counted and then pinpointed with their locations on the slide. These pinpointed fragments were then exposed to Raman spectroscopy to determine the composition. I did have some trouble using Raman spectroscopy because it's typically used for geological purposes for organic matter, and I was having trouble matching anything with the samples in the database. And so I then brought my samples over to HPU to use their Fourier transform infrared spectroscopy, which is a better application for this project because it's most commonly used for synthetic materials such as plastic. And it also contains a fairly comprehensive spectral library of plastics as well as of weathered plastics. This is the suspected plastic fragment count by size classification. So this is before any further spectroscopy analysis, just based on visually identifying. As you can see, there's a significant amount of fragments visually identified, and the 200 micron size classification is dominant. This is the analysis from FTIR of the most commonly seen fragment, and this fragment made up most of all of these fragments that I counted. And as you can see in the table below, you have the compound name on the right, and then just to the left, you have the match. So a match of 70 or above is considered significant. And if you look at all of the compound names, you'll notice that it's all organic matter. You have pine, dried leaf, some seeds, things like that, and none of it is synthetic, so none of it's plastic. So the image on the right is that most commonly seen fragment that I had visually identified as plastic. But overall, the fragments that were thought to be plastic based on morphology were identified as organic materials. It's likely that these organic materials survived the wet peroxide oxidation due to their thick cell walls, whereas other materials such as plankton were more easily dissolved. I did implement a control here. I was a little bit worried that my sample processing technique was maybe dissolving the plastics from the plankton toes. So I used virgin polypropylene and polyethylene from store-bought materials. I cut them into 1 millimeter by 1 millimeter squares and inserted them into a couple of my samples. And I processed those like I usually would. After that, I took those squares of polypropylene and polyethylene and examined them under a microscope to look for any signs of degradation, and there was none. So it's likely that the lack of plastics is not due to any errors in the sample processing. So overall, I did determine a lack of plastics detected in the surface waters of the Kāneʻohe Bay sampan channel. These are relatively surprising results and not what I had hypothesized, and there are a few potential explanations for a lack of plastics within this area. The first is that growing evidence is showing that plastics are typically found deeper in the water column. So maybe if I had done my plankton toes a little bit deeper, past that 0 to 1 meter depth, I may have found more plastics, though this doesn't typically apply to polypropylene and polyethylene, as again, they do tend to float. It's also possible that plastics may reside more inside of the bay, closer to the populated shoreline. So maybe if I had conducted my toes closer to the shoreline, I may have run into more plastics. Another potential explanation is that plastics may accumulate in slicks outside of the bay and wash through the channel in intervals, and perhaps I missed those intervals when the plastic was traveling through the channel. And then another potential explanation is that circulation patterns may just aggregate those plastics in an area away from my sampling location. I want to thank Kayla Brignac at HPU for her consistent advice and allowing me to use her FTIR. Professor Shiv K. Sharma for allowing me to use his Raman spectroscopy lab. Dr. Karen Self for allowing me to use her microscope lab. Thank you to Dr. Margaret McManus and Dr. Jonathan Whitney for helping me interpret the results of this thesis. Noah Howins for his constant advice and help throughout the process, and Dr. Christopher Sabine for mentoring me through this. I also want to thank the Pacific Islands Climate Adaptation Science Center for funding me through the Summer Undergraduate Research Fellowship, as well as Europe for their funding as well. Any questions?
Great, thank you. So, any questions?
Hi, I have a quick question. I was wondering, what was the main reason why you chose to sample in Kāneʻohe Bay?
Yeah, that's a great question. So Kāneʻohe Bay is the largest sheltered body of water within the main Hawaiian Islands. And it's also very populated. So it's unique in that it's a very populated shoreline, as well as it's right next to the Marine Corps base there. And there's a lot of boat traffic that goes in and out. So I was expecting to see a relatively high level of pollution with plastic in that area. I also hadn't seen much research done on microplastics within Kāneʻohe Bay. So I did want to explore that.
Thank you. Great talk.
Thanks.
Hi, Samantha, I have a question.
Yeah.
So is there, or did you decide to, or what was your reason behind looking at microplastics that were less than 1 millimeter? Did you think about looking at plastics that were between 1 millimeter and 5 millimeters? Or did you look at that?
Yeah, I was also going off of the NOAA methods. The paper that I was going off of, that is their size classifications that they used. I was thinking that I could, increase those size classifications if I ended up actually visually seeing plastics in the surface waters, and I ended up not seeing any.
Oh, that's interesting. Great job. Thank you.
Thanks.
Hi, Samantha. It's Jeff. Really nice talk. Really surprising result, as you indicated. I'm just curious if you've looked at circulation patterns in Kāneʻohe Bay and sort of tried to make any predictions about where these other locales might be accumulating plastics.
Yeah, so that's actually what I've been working on a little bit. Kind of now I've been looking into the surface currents in Kāneʻohe Bay. I haven't found any dominant currents quite yet within the bay, but that is something that I would recommend for future research and that I've been looking into.
Cool, thanks.
Hi, Sam. Can I ask you a quick question?
Yeah.
If you were to take this one step further, or if someone was to pick up your research, what would be sort of your recommendations for the next step in locating microplastics around the Hawaiian Islands?
Yeah, so I would say, within Kāneʻohe Bay specifically, like I referenced here, I would talk about, it's possible that within closer to the shoreline or other areas of the bay, in case that there are plastics that are aggregated in different areas, I would also look at those surface currents more and see if there is any indication that there would be, accumulation of plastics in certain areas. I would also say it would be helpful to sample a little bit deeper in the water column and see if you come up with anything there, as well as sampling with more weather variability, because there was no, significant rain or anything like that between my sampling. And I think that it would be interesting to look at, any storm events and how that affects the concentrations there.
Samantha, this is Jim, as long as I got a second here. Yeah. There is also the possibility, of course, that there just aren't any microplastics in Kāneʻohe Bay, right?
Right. Yeah, that would be surprising, but yeah, it's a possibility.
Right. I think actually you're right. The work that was done out at some of the Northwest Pacific Islands, your point number 3 here, I think is really the explanation that these things are in some sort of reservoir. And there's these extreme events, probably wave or wind events, that push it over the reef into the onshore environment. So, yeah. So I think number 3 is really where we're headed. Thanks.
Yeah.
Okay, great job, Samantha.
Thank you.
So our next presenter also is mentored by Kasey Barton, who is also in Europe at the moment. She's in the Botany department. She sent a paragraph for me to read to introduce our next speaker, Devyn. So it reads, I regret that I'm unable to introduce Devyn Montesinos myself, but I am happy to be able to share a few words about her project with me. It is not that common for students to pursue independent research in a different major to the one they are pursuing. But it's always a pleasure to mentor a student with the drive and curiosity to explore new fields. For her GES thesis, Devyn conducted a series of experiments to test whether coastal dune plants can survive and recover from simulated flooding due to sea level rise. While many studies have tested the tolerance of dune plants to chronic exposure to high salinity, Devyn's is the first to test how complete inundation affects dune plants in Hawaii. To successfully run these experiments and collect the data, Devyn learned many new skills, including horticulture, greenhouse maintenance, basic seawater chemistry, plant physiology, trait analysis, data collection, and analysis. Not only did Devyn succeed in all these new skills, she conducted these experiments with precision, attention, and hard work to be able to produce some really exciting results. It has been a pleasure to work with Devyn, and I look forward to seeing where she casts her grit and enthusiasm next. So, Devyn.
Thank you for the introduction. Aloha, my name is Devyn, and I have been dealing with COVID for the past couple of days, so please excuse my voice. I'll try to be as clear as possible. But I did my GES undergraduate thesis on Hawaiian dew plant tolerance to simulated coastal flooding. My mentor is Dr. Kasey Barton of the School of Life Science, and my project is in collaboration with the University of Hawaii Sea Grant Program. So I'm working with the plants that grow in the coastal dune ecosystems, and these are really beautiful habitats that live right next door from the ocean and host a diverse amount of plant life. And even though they're small, narrow ecosystems, they play an extremely important role ecologically, economically, and culturally. And these very small systems are actually very heavily stressed, and they're not an easy place for a plant to grow. And some examples of this are listed below. I wanted to take a minute to talk about the ecohydrology of these ecosystems, since coastal dune communities do occur above the high tide zone, making them freshwater systems. They do depend on their roots to dig down and to access fresh groundwater, vital for their survival. And because of their close proximity to the ocean, represented by this star here, they are regularly exposed to salt stress, and this can occur above ground by salt spray, from the wind and large waves, wave run-up from storm events, and also below ground from saltwater intrusion. And this issue has actually become more pressing under sea level rise. The coastal ecosystems are at risk from anthropogenic climate change, and it is predicted that there'll be a higher frequency of extreme weather events that can lead to intense seawater flooding. And this is exacerbated by the rising sea level. Experts are also projecting reduced rainfall, which can lead to a greater accumulation of substrate salts. The combined pressure from human activities and densities in coastal dune habitats, plus the climate change exacerbating natural stressors, are making these environments more stressed than they have historically. Increased salinity can affect a plant two ways, via direct toxicity or as ionic stress, as well as osmotic stress. So ionic stress occurs within the cells when accumulated salt ions disrupt membrane stability, protein structure, ion transport dynamics, while reducing photosynthesis, protein synthesis, and enzyme activity. Osmotic stress results from high concentrations of salt in the soil, which reduces soil water potential and constrains water uptake by the roots, while reducing cell elongation and leaf development. Also, plants exposed to this coastal flooding will experience oxygen limitation due to the lack of gas exchange in the roots, and you'll see this is why I added a freshwater treatment to my experiment. However, both of these stresses can reduce photosynthesis and growth and potentially lead to plant death. So plant species vary considerably in their salinity tolerance, ranging from high tolerance in halophytes and low tolerance in glycophytes. Strategies in salinity tolerance include mechanisms associated with osmotic stress tolerance and traits that limit the salt ion uptake or minimize cellular toxicity. Salinity tolerance varies not only among species, but also within species as regarding to their plant age and ontogeny, environmental conditions, irrigation management, soil fertility, and the intensity of other stresses on that plant. Previous studies have generally recognized that seedlings have a higher mortality rate than any other life stage, and this can affect seedling recruitment since it's essential for population dynamics and stability. Climate change has the ability to drive plant population declines through the specific suppression of seedling recruitment since there are more vulnerable. This is leading us to my main question of my experiment: how can coastal dune plants grow and survive under high salinity under simulated coastal flooding? And to investigate this, I conducted a controlled experiment to test the physiological and growth responses to seedlings to short-term simulated water undulation. I did this on 4 common and widespread native and non-native species. First, we have the Josiphium, also known as ma'au, and this is an endemic shrub. And then we have the Witheria, also known as the ualoa, and this is an indigenous shrub. And we have the Propus, also known as kiave, and this is an invasive noxious weed. And then we have the Bidens, and this is an invasive touch. Oh, excuse me. Seeds for, this is actually how I tested salinity tolerance in these seedlings. So seeds were scarcified and germinated under greenhouse conditions for about 2 weeks. And after that, they were transplanted into 1-gallon pots and were grown for 2 more weeks before treatments were initiated. These seedlings are randomly assigned to 3 treatment groups: salinity, freshwater, and control. And the indonation groups were immersed just below the top of the pots in large plastic tubs with a 34 ppt instant ocean solution for 12 hours. 34 ppt is the typical concentration of seawater, so we tried to make a mimic of it, and this concentration was confirmed using a salinity meter. The control groups were not inundated, and then immediately after inundation, the plants were randomly arranged back on the benches to allow for free drainage. During the 3 recovery weeks, plants were watered daily with fresh water while growing under the greenhouse conditions. And then following that recovery period, plants were harvested to allow us to access the growth of plants in response to those treatments. And again, by adding a freshwater treatment, I'm going to be able to tell if these effects of the coastal flooding are due to the lack of oxygen or due to the lack of oxygen and salinity stress. So I had a sample size that included 10 replicate seedlings per treatment per species, producing a total sample size of 120. However, because of mortality from transplant shock, it reduced my total sample size to 115. On the left here, this is a barometer. This allows us to measure the model conductance, which is gas exchange, and this is a good way to understand plant response to water limitations. And it's our approach to measure the osmotic stress component. And then on right next to it, we have this SPAD machine, which is measuring the leaf chlorophyll content, which is going to help us understand whether there could be ionic stress happening to the plants, which will then manifest itself as a reduction in enzyme function. These photosynthetic traits are measured weekly, starting 1 day before inundations, and these tools allow us, informs us of how salinity stress occurs. Height and biomass were also measured at the end of the experiment, while survival was measured throughout. And down here for the interpreting responses, these are the general patterns found to help interpret our results, and this will help in the next couple slides. So these survival rates varied considerably across species in the seawater treatment, and we can see that since we have a range from 0 to 100, indicating there is a large variation in salinity tolerance. And specifically, there was no mortality observed in the Propus, indicating full tolerance, while there was 100% mortality observed in the Bidens, indicating that they had no salinity tolerance at all, while the 2 native species experienced intermediate tolerance. And across all species, no mortality occurred in the control or freshwater conditions, revealing that flooding alone does not only drive mortality, and it suggests that high salinity is responsible for the low survival in seawater indonation groups. And here on the graphs on the left here, these are the group means of growth in terms of total dry biomass and height, and I'm showing you the different species in the growth of plants from the controlled freshwater and seawater treatments. I don't have the Bidens here because of technicalities. I was not able to get the biomass for it. But if you look down here on graph B in the Propus, this is what full tolerance should look like: full levels of growth under all treatments. The Propus was fully able to tolerate both treatments with respect to height, but we do see a reduction in biomass kind of suggesting that they are growing as tall, but they're not as able to put that much weight on. The gossypiums had a similar pattern with a slight reduction in growth under seawater inundation, but we are seeing extremely low tolerance in respect to growth with the witherias, with massive reductions in height and biomass. And in general, freshwater inundation did not negatively affect plant growth. The witherias even appeared to grow better compared to its control. On the table on the right is for your reference to look at the statistics for the statistically significant results. And here we have the stomatal conductance, which represents whether or not the stomata are actually open, and that is something the plant can regulate when they're experiencing water stress. We measured this repeatedly over time, and each of these points are showing you the responses within treatment groups over the course of the whole experiment. And we have control in this dotted line here, freshwater in this light gray, and the black line is the seawater. In graph A, in the Bidens, you can tell this one stops about right here on day 22, just because this group experienced 100% mortality. Now, so you can see in the beginning, a lot of these, excuse me. In this beginning, we see similar conditions, but after the inundation, we start seeing these massive reductions because they're starting to close their stomata because of water stress. And the reduction is quite consistent across the species. However, the magnitude of those reductions are variable. But the key here is that some of these species were able to recover that stomatal conductance back to similar levels before inundation. And that is what we see for the Propus, and not to the same level, but more or less with the Josephium. But the Witherias here, which if we remember was the species with the massive growth reductions, we can see that the stomatal conductance remains low even after 3 weeks of a freshwater recovery. We can actually see why these plants aren't growing. They close their stomata to conserve water, but at that constraint are no longer taking CO2 for photosynthesis. And here are the chlorophyll content. This is an important synthetic pigment to the plant, largely determining photosynthetic capacity, hence plant growth. And we measured again this repeatedly over time, and these points are just showing you the response within treatment groups over the course of the whole experiment. Similar temporal patterns were detected for leaf chlorophyll content, confirming the recovery of the Josephium and the Propus, but not for the Witheria and the Bidens. In kind of summarizing my main results, I found that salinity tolerance is highly variable across species, kind of revealing a complexity within coastal dune communities in their response to sea level rise. Inundation stress. Also, I found that inundation stress is due to salinity and not oxygen limitation. Since freshwater inundation did not negatively affect any plant growth or photosynthetic traits, the principal effects of the immersion in seawater were due to solely salinity stress. Also, I found that the recovery following inundation depends on the recovery of stomatal conductance from my data. Also, I found there was no clear difference between natives and non-natives. While my species pool doesn't allow for a robust contrast of salinity tolerance native versus invasive species, only because 2 of each were tested, there was no indication that invasives were more salt tolerant than native species. So my project is actually a part of an ongoing larger project with the University of Hawaii Sea Grant Program, with the principal investigator being Kasey Dr. Barton herself, and some co-investigators from the National Tropical Garden and the Maui Nui Botanical Garden. So the Barton Lab is proposing to develop an online public database to make our data and results widely available to practitioners who are working in conservation of these systems, or anyone that might be able to use this information of salinity tolerance or future research that will improve our understanding of coastal dune ecology and its potential resilience. Climate change's projections highlight the urgent need for this information about species resilience to salinity stress, and my study can help inform management and conservation of Hawaii's coastal dune plant communities. Considering my experimental conditions, I found several limitations to these results. First, time was limited, so relatively few species were tested over short durations. Running longer experiments may alter their conclusions. For example, some species were able to tolerate this salinity over longer periods of time. Also, the use of instant ocean solution rather than actual ocean water However, I found previous salinity Indonesian studies reporting that there was no detectable differences between instant ocean and seawater, providing some justification for that. Lastly, this experiment solely isolates the effects of indonation and does not account for the greater complexity conditions of the natural habitat where this coastal flooding will occur. Climate change is a complex multidimensional force, and in reality, plants are not only coping with salinity effects, but also simultaneously with heat and drought stress, as well as other biotic interactions. Future research directions could expand by experimenting across ontogeny, so we can have a more accurate picture of how these species will react under seawater indonation, and also by testing a broader diversity of native and invasive species will provide stronger evidence for whether salinity tolerance differs between native and invasive species in Hawaii. Overall, these future research directions will improve our understanding of coastal dune ecology and potential resilience under climate change, and by providing insights into the future of these critical ecosystems and enhancing their conservation. I'd just like to acknowledge my thesis mentor, Kasey Barton, who helped guide me through this entire process. I couldn't have done it without her. Also, I'd like to acknowledge Anna McCormick, who was Kasey's graduate research assistant, who allowed me to shadow her during her experiments. And while I express my gratitude to the Oceanography department and my friends and family. Thank you. Any questions?
Nice job. I think we have time for one question. Put the hand up there. Petra?
Yeah, so what other species would you suggest would be high priority for testing?
That's a really good question. I would say looking into invasive species just because they could outcompete and outgrow with native species. And what I found with the propus was that it was actually very salt tolerant, and that could be a danger in these communities if these sea levels start rising and more salt accumulation starts happening and these invasive species are kind of spreading out and the native species are dying out. So I think it'd be really interesting for high priority to test more just native and culturally important native species and invasive species that may pose as a danger to them.
Thank you.
Thank you.
Great, thank you. We'll have to move on now. So our next presenter will be introduced by Dr. Robert Richmond of Koalo Marine Laboratory.
Thanks, Glenn. Good morning, everyone. It's my pleasure to introduce Lauryn Hansen, who will present on her work on PFOAs. Lauryn grew up along the west coast of the US, along Puget Sound, and later along the coast of Southern California, which really piqued her interest in coastal marine communities. She indicated that when she was in high school, she really got excited about environmental science and decided that this was the path that she was going to pursue. And when she first approached me about working in our laboratory on some of her work, she was just a perfect fit of work we do on ecotoxicology, what we affectionately call Dr. Doolittle meets CSI on a coral reef. And she's been an outstanding member of our lab and being able to do what we call the so what science. She presently is working on a master's degree in public health. And this is particularly neat to see her understanding and appreciation for the very strong links between environmental health and human health. And so it's a pleasure to introduce Lauryn to talk about her very interesting work on PFOAs. Lauryn?
Thank you so much, Dr. Richmond. Yeah, my name is Lauryn Hansen, and my title, my thesis presentation is the impacts of perfluorooctanoic acid exposure on Hawaiian corals. And my mentor was Dr. Robert Richmond of Koalo Marine Laboratory. There we go. So coral reefs are incredibly valuable both to coastal communities and globally for their various ecosystem services. They're one of the most biodiverse ecosystems on the planet. They host about a quarter of the world's marine life, which makes them a natural source of food and natural product supply to nearshore fisheries, and they can also generate revenue through ecotourism activities. They're also the primary line of defense in coastal protection. They prevent damages from damages to coastlines from sea level rise and storm surges, and also have incredible cultural value in the Hawaiian Islands specifically. In the Kumalipa or Hawaiian sacred creation chant, they're depicted as the first living organism on the islands and therefore one of the first ancestors of the Hawaiian people. However, coral reefs are subject to a series of anthropogenic or human-induced stressors, including marine pollution, which can decrease the water quality surrounding coral reefs and ultimately interfere with their reproductive success and also cause metabolic stress in adult corals. Additionally, climate change and ocean acidification also cause larger global threats to coral reefs, such as bleaching through the increased frequency and intensity of thermal events by climate change. Coral bleaching is the expelling of the zooxanthellae algae that provides corals with the majority of their nutrients. And ocean acidification can also inhibit the ability of coral reefs to develop their skeletons and can ultimately lead to more brittle coral populations that are vulnerable to external stressors such as marine pollution. The chemical that I'm focusing on in this experiment is perfluorooctanoic acid, or PFOA, which is one of the most commonly detected species of per and polyfluoroalkyl substances, or PFAS chemicals. This is a group of man-made fluoropolymers that are primarily used for their water, oil, and stain-resistant properties. They're used in commercial and industrial products ranging from cookware, textiles, firefighting foams, to paints, personal care products. The list really goes on. And because of the strength of the carbon-fluorine covalent bond and their fluorinated alkyl chains, PFOA and other PFAS chemicals are essentially resistant to any form of environmental degradation. And because of this, have been found ubiquitously around the globe, from ocean sediments to marine environments globally, even as far as the remote polar regions. And PFOA and other PFAS compounds can reach the natural environment both directly from their manufacturing facilities and through landfill leachate and runoff. And they've also been detected in the blood of almost all humans and animals in these developed areas, and have been found in select studies on humans, rats, and select marine mammals to be an endocrine disrupting compound. They've had effects ranging from decreased fertility and thyroid disease to tumors of the liver, testes, and pancreas, among other health implications. However, no study has yet quantified the impact that PFOA has on coral reefs. And therefore, the question that I'm trying to answer is, how does PFOA exposure influence various phases of the coral life cycle on Oahu? And I attempted to answer this question in 4 parts. I looked at fertilization success with exposure to PFOA, larval survival rates, physiological observations of stress with adult exposure periods, and also looking at molecular indicators of stress by doing cellular diagnostic techniques after these adult exposures. I ultimately decided my exposure concentrations based on the EPA health advisory level, which is 70 parts per trillion, and also using real world ocean sampling data from the Pacific Ocean and around the Hawaiian Islands. I ultimately ended up deciding on 0.01 parts per trillion, 4 parts per trillion, and 142 parts per trillion as the remaining concentrations, with filtered seawater as our control. And for our fertilization assays, we performed 5 collections of Montipora capitata, or rice coral gametes, from Kāneʻohe Bay. One of these collections, we exposed the gametes to concentrations in parts per billion, or 1,000 times stronger than our original concentrations. And this is just to account for accumulation of PFOA in the natural environment due to their lack of degradation mechanisms. And for each of these collections, we scored fertilization success after 5 hours, or at the 16-cell stage of development. And as you can see in this little image I attached to the right, we determined fertilization success. An unfertilized gamete under a light microscope has this spherical shape, whereas a fertilized gamete will have this clover-like structure. For the larval survival portion of my experiment, I performed 2 experimental replicates. Both of these we grew excess larvae from our Montipora capitata collections. And we waited until they were about 9 days old and selected about 10 larvae per treatment. I placed them in these small little jars here with the different concentrations, and quantified their survival rates after 24 and 48-hour incubation periods. When I repeated this experiment, I added 1 additional concentration of 1,000 times the EPA health advisory level, or 70 parts per billion, and I quantified those survival rates after just 48 hours. For our adult exposure experiments, we did 3 and 7 day exposure periods with Carides lobata, or lobe corals. And the reason we switched our study population for this portion of the experiment is because As opposed to M. capitata, lobe corals tend to be a little bit more resilient to external stress. And so we used them as a better indicator. We observed each day for observations of bleaching, presence of polyps, and any production of mucus. We measured bleaching using the Hawaiian Koa card, which is featured in the corner of this little image here with all my coral samples. And after completion of the exposure periods, we performed SDS phage and Western blotting techniques to determine any sublethal indicators of stress. And how we did this was we extracted the proteins from these samples, inserted them into these polyacrylamide gels to separate out the proteins, transfer them to a membrane, and applied 6 different known molecular biomarkers, and then compared the net intensity of the band signal between these visualizations. In our fertilization assays, we did see some visible decreases in fertilization success. However, the results were statistically insignificant, and they were also inconsistent between collection nights. The inconsistency is likely due to just natural variability in gamete condition. But I did go ahead and attach the most clear indication of decreased fertilization success that I saw with exposure to PFOA. And you can see in this graph here, the median fertilization rate does decrease with increasing concentration. However, there is high variation, especially in our higher concentrations. And so we are unable to conclude with absolute certainty whether there is a deleterious effect of PFOA exposure on fertilization success. However, I also cannot say that there is no effect. In our larval survival experiments, even when we added a concentration that was 1,000 times the EPA health advisory level, we still saw no significant difference in larval survival rates. And you can see from the control to the largest concentration, those survival rates remain relatively consistent. During the adult exposures or physiological observations, we saw slight indications of bleaching in some of the coral samples. However, it was very minor, and we also had no significant mortality events during these exposure periods. In terms of mucus production and polyp presence, there was no mucus production found in our samples, and polyp presence varied, but it wasn't necessarily by treatment. It was just kind of variable by the samples themselves. What was more interesting is 2 of the 6 molecular biomarkers that we used did show signs of upregulation. And these 2 were glutathione reductase, or anti-GSR, which is used to measure signs of oxidative stress. And you can see here, especially in our 142 parts per trillion group, in the longer term exposure, we have an exponential increase in net band intensity. And with cytochrome P450, which is used to measure signs of detoxification, we see a more linear increase in our 7-day exposures. However, again, in the 142 parts per trillion 7-day exposure group, that is when we see our most significant upregulation. And this essentially just tells us that with higher concentrations and longer exposure periods, we will see molecular impacts on corals. Going into our discussion, the reason that we likely didn't see any significant impacts on survival and fertilization success and, larval survival is as opposed to other endocrine disrupting compounds, which tend to be lipophilic in characteristics. PFOA is lipophobic with protein binding characteristics, and gametes and planiella larvae are composed primarily of lipids with low protein content. So it does make sense why these short-term exposure periods wouldn't have a significant effect, especially on larval survival. However, like I mentioned earlier, there still is potential to impact fertilization success. Just because I didn't find statistically significant results doesn't mean that they wouldn't be found in a replicate study. With our adult exposures to PFOA, we did see the upregulation of those biomarkers involved in xenobiotic metabolism or detoxification and oxidative stress. And what that tells us is as these corals are reallocating their energy towards stress response, they're taking away energy reserves for reproductive success. And so that can have potential implications on long-term fertility of these organisms. And I went attached, this is from one of our gamete collections here. These are just some of the gamete bundles that we got from Kāneʻohe Bay. And this is an image of a healthy Parietus lobata coral surrounded by some bleached Montevera capitata. Moving forward into future studies, making my exposure concentrations, we did have to perform multiple dilutions. Parts per trillion is a very small concentration, and so having to perform these multiple dilutions does decrease our confidence in the actual concentration amounts. So using detection technology in future experiments would be extremely beneficial to increase our confidence in our results. In addition, in our Western blot process, we had to use polyclonal non-specific antibodies. Using cnidarian-specific antibodies in future studies would have more effective specific binding and provide us with more accurate results. And additionally, using more specific proteomic analyses in future studies would provide us with a fuller picture of PFOA impacts on coral reefs. For example, using LCMS-MS would allow us to map the entire metabolome profile of these organisms, and we would be able to see the specific impacts PFOA has on different metabolic pathways. Additionally, looking at impacts on long-term fecundity of these corals, as I mentioned, the impacts on oxidative stress and detoxification can have long-term implications on reproductive success. So future studies should perhaps shift their focus to that. And also, one study that I looked at did find metabolic impacts on plants with exposure to PFOA. So looking at if this can be extended to the zooxanthellae algae within the coral organism would be also beneficial for future studies. And to conclude, PFOA does still pose a threat to both coral reefs and public health, but further studies do need to be performed to quantify the deleterious effects that it has. Coral reefs are still incredibly vulnerable to changing global conditions, but by decreasing the impact that land-based pollutants have on coral reefs, we essentially buy time to preserve these reef ecosystems from these larger global stressors we have less control over. And with that, I would like to thank my family, peers, and Dr. Richmond for their support and for constantly encouraging me to challenge myself academically. And an extra special thank you to PhD candidate Alexandria Barkman. She put in hours of time with me throughout this whole process, and I really couldn't have done this without her. So with that, any questions?
Okay. Nice job. We have time for one question.
Hello. I have a question about what you said about the antibodies in the coral. So I'm sorry I jumped in the presentation a bit late, but what would be the application of looking at antibodies for this project specifically? Could you clarify?
Yeah, so the antibodies we use are clinical antibodies. They're typically used to see if you have any type of metabolic stress in an organism, and that can be in response to anything. Like oxidative stress, for example, is also a stress response to coral bleaching. And so basically, we're just trying to determine if there's any impacts that we can't see with the naked eye in these coral organisms. I don't know if that answers your question specifically, or if you were asking more which ones we were using.
Oh, no, that's good enough for me. Thank you very much.
Okay, perfect, thank you.
Okay, nice job. So moving on, our next speaker will be introduced by Dr. Michael Cooney of the Hawaii Natural Energy Institute.
Hello, I'm introducing Seraphina, who is a GES student who came from Hilo as a transfer. And soon afterwards, Seraphina decided that dental school was in her future. So she seeked out a project that bridged the two fields. And that's what she's going to present today. Currently, Seraphina is working as a nurse's aide to get experience before she starts dental school in 2023. So, Seraphina, it's all yours.
Thank you, Professor Cooney, for that introduction. I appreciate it. So yes, I'm Seraphina, and today I'm going to be discussing my research thesis on the assessment of methods of fluoridation. Okay. Yeah, so my research thesis was on the assessment of methods of fluoridation to conclude the most beneficial for human health and the environment. So just a little bit of a table of contents debrief on what we're talking about. First, I'm going to be going through my introduction, giving you guys a little bit of background information on dental fluoridation. And then I'm going to be going into my methods, going in depth analysis on my results and discussion, and last but not least, my conclusion. So for my introduction, first, yes, I'm going to be talking about dental fluoridation. What exactly is it and how is it used? What are its benefits? And then I'm going to be talking about the 8 methods of fluoridation that I chose for my thesis and for my analysis. I'll go more in depth about typical and systemic in just a bit. So for my introduction, dental fluoridation has been proven to improve the condition of a person's teeth by strengthening the enamel and preventing cavities from occurring. Fluoride is applied in various different methods to achieve maximum oral health benefits. As you know, oral health is a big indicator of a person's overall health. It's very important. So the methods of fluoridation, so on the left-hand side, we have systemic, which is meaning that fluoride is going to be directly ingested into the body. The 3 methods that I chose to analyze are going to be municipal water, bottled water, and fluoride supplements. On the other hand, we have topical, which means that the fluoride is going to be directly applied to the surface of the teeth. And I'm going to be analyzing mouth rinse, toothpaste, varnishes, gels, and foams. Going next, so now we're going to be talking about the methods. So for my methods, I'm going to be doing, I did a sustainability analysis. With the 8 methods previously mentioned, I decided to get 9 impact categories that were selected under the 3 pillars of sustainability. So the impact categories for environmental, I decided to use were impact on environment, toxicity, plastic pollution. For economic, I decided to use recyclable, costs, and research consumption. And for the social pillars, I decided to do health risks, health benefits, and participation. So each of these 9 impact categories were scored on a scale of 0 to 5, 0 meaning unsustainable and 5 being extremely sustainable. So next, this is just an example of how the impact categories were scored in depth. So on the left, I have the methods of fluoridation, and this is specifically for the impact on environment. So I was analyzing what form of fluoride is used per method, the life cycle starting from cradle, usage, and ending in the end of the life cycle. And then I was able to come up with an analysis on the environmental damage to come up with my score for that impact category from 0 to 5. So continuing, so I'm going to be going into my results and discussion. I decided to do a radar plot analysis in order to have a visual component for comparison for my methods. So here we go. So here are my first 2 methods, mouthwash and toothpaste. As you can see, 0 is in the middle and 5 is on the outer skirts of the radar plot. So the more coverage per plot, the more sustainable the method is. So I also subdivided it in groups from environmental, economic, and social, so you can see if any of the methods have more favoritism towards one of the pillars of sustainability. So here, mouthwash and toothpaste tend to have like average coverage, not too high, but not too low. Continuing with gels and varnishes, as you can see, the plots seem to be very similar. Professionally applied fluoride treatments like gels, foams, and varnishes tend to have higher concentrations of fluoride. So as you can see, varnishes is the highest. It has a 0 score of sustainability. It has a ppm of 22,400 ppm. So continuing, as you can see, foams are the 3rd fluoride treatment, and they are also, it has a similar plot as well. On the right-hand side, you can see the municipal water fluoridation, that the coverage is a lot more, and you can see that a lot of the impact categories were scored very highly as a 5. So yes, I'm gonna go more in depth than that. So here we have the bottled water and the fluoride supplement radar plots. As you can see, there's more of an average sustainability score for these 2 plots. So here we go. So after getting all of the scores for my 9 impact categories, I was able to combine them and rate them on a scale of 1 to 8, 1 being the most sustainable and 8 being the least. So as you can see here, municipal water scored a 39, which gave it the most sustainable method. And then leading with toothpaste, mouthwash, fluoride supplements, bottled water, gels, foams, and last but not least, varnishes at a score of 19 of sustainability. So now the comparison and discussion. So most sustainable versus least sustainable. So municipal water fluoridation coverage was higher for all of the pillars of sustainability compared to the other plots. We concluded that there was little to zero environmental impact because it is directly applied at water treatment plants and therefore produces no waste and is at no cost to the consumer. The health risks are low since the toxicity of municipal water is only at 0.7 milligrams per liter. So the only risk that would come from this method would be overconsumption with the combination of other methods of fluoride. So participation is determined by local officials who decide whether or not to fluoridate public water supply, and if so, how much fluoride to apply to the water. But on average, 207.4 million Americans consume municipal water daily, which is approximately 70% of the US population. There is no concern regarding municipal water's impact on the environment because the quantity applied isn't significant enough to have an impact on the environment, along with dilution from other runoff sources. And no health concerns were noted besides if it were to be overconsumed over a long period of time with other methods of fluoridation. So on the right-hand side, you can see varnish, least sustainable. The coverage is very low. There was no favoritism for any of the pillars of sustainability. It's not recyclable, and it's mass manufactured, which damages the environment. Cost is higher at an average of $20 to $50 per treatment. The health risks are low due to the fact that it's professionally applied, but the toxicity is extremely high at a concentration of 22,400 ppm. And the participation is average since dental professionals use it at their discretion and if needed. So varnish fluoride treatments are beneficial to human health by preventing tooth decay, but specifically for people who lack fluoride from other sources of, from other methods. So going on to my conclusion, so municipal water is the most sustainable method of fluoridation and poses no concern to human or environmental health unless it is consumed over a long period of time along with other methods. Fluoride varnish is the least sustainable method of fluoridation, specifically due to low participation and high toxicity. But the takeaway mainly from everything is that the combination of brushing regularly with toothpaste 1 to 3 times a day, drinking from municipal water on a regular basis, and using fluoride treatments only as needed by dental professionals is going to be the safest way to receive benefits of fluoridation, as well as protect human health and the environment. So, conclusion. So, welcome for any questions, and thank you so much for listening.
Okay. Thank you. Nice job. Any questions? So do all regions of the US have fluorinated municipal supply, or?
Sorry, only about 70% of the US fluoridates their water. Places like, actually Honolulu doesn't fluoridate their water, and it depends on the public officials. It really isn't up to the vote of the people. It's up to public officials that decide whether or not, and if so, the quantity of fluoride that is applied. And it would be done in water treatment plants. That's where the fluoride would be applied.
I have a question, Glenn.
Yes.
Nice job, Stefania. So in approaching and judging sustainability of a process or a product this way, where you look at the 3 pillars, scientists are generally used to only weighting science, or in this case, environmental impacts, like that's all they weight, that's all they focus. The approach you took considers economic interests and social interests as well. But the social interests can be pretty hard to score because they're very qualitative. So can you give any overview or perspective from your point about how you went about judging or scoring the social impact categories that you chose?
Yes. So I decided to use, so for the 3 pillars of sustainability, the social one was a bit harder to find the impact categories that would be the most sufficient to provide the best data for my analysis. But I chose the health risks, the health benefits, and the participation. The participation was the first one I came with because obviously the amount of people that are using the method is going to have a huge factor in my analysis. But the health risks and benefits, I just decided that it would be best, since my my assessment is on human health and the environment, I wanted to make sure to include some aspects that would have to do with human health and if the method is potentially harmful. So that was why I chose health risks and health benefits as well as participation. I just wanted to include some health aspect of my analysis, and I chose to do it in my social component.
Okay, so I get that, and that's justifying how you selected them. What I'm curious about is what was your approach or your thoughts towards how to score them between 0 and 5? Because that's qualitative, and you know, in science you can get numbers and you can get a baseline.
Yes. Oh, okay. I understand now. Sorry about that. So yes, I was able to locate primary and secondary sources, specifically mostly using the ADA, the American Dental Association, the CDC, Centers for Disease and Control, and everything. So I was able to use those primary sources to analyze and choose. Sorry, one second. Sorry. Yeah, I was able to figure out how to score each of, I was able to get the data that I needed to score my impact categories from the primary sources, the ADA and the CDC. So I was just able to get a thorough analysis and more data on each of the impact categories so that I was able to score them on a score of 0 to 5.
Okay, thank you.
Thank you.
Great, thank you. Our next presenter will be introduced by Dr. David Bielman from Geography and the Environment.
Great, thank you. It's my pleasure to introduce to you Ms. Tiana Hughes. Tiana is born and raised right here in Oʻahu and graduated from St. Andrew's Schools in 2018 and has gotten all the way through her GES degree right on time. Well done, Tiana. And Tiana started off with an interest that started in environmental science, kind of took her to policy questions and then back to more quantitative science-related questions when she came to my lab. And I originally hired her as a laboratory assistant. And that went really well. And I said to her one day, hey, Tiana, why don't you think about nitrogen cycling in Antarctica? And she took the bait and turned it into a great UROP and GES project. And the final result kind of speaks to her interests in marine science and this interesting case study of how that fits in with connections between the marine realm and terrestrial ecology. Thanks, Tiana.
Thanks for that introduction, Dave. So once again, my name is Tiana Hughes. My mentor is Dr. David Bielman, and my GES thesis is titled Using Nitrogen Stable Isotope Values to Track Nitrogen Cycling Over the Last 4,000 Years at Cape Tuxin, Antarctic Peninsula. So just to give a little background about climate change in Antarctica, climate change and rising temperatures, as we know, are causing significant ice melt in the region, and this is causing an opportunity for colonization by land plants. And the Western Antarctic Peninsula in particular is one of the most rapidly warming regions on the globe. And on the figure on the right, we see an air temperature anomaly in the summer of 2020 and the Antarctic summer of 2020. And here, a record-breaking temperature of 20.75 degrees Celsius was recorded on the Antarctic Peninsula. And cross-hatching marks regions where the anomaly exceeds the climatological maximum. So going into how this is causing greening, the most dominant types of plants here are bryophytes, which are essentially the group name for mosses, liverworts, and hornworts. They are non-vascular plants, which means they absorb water and nutrients from the air through surface tissue. And for the purpose of our study, we're focusing on Polytrichum strictum, which is also known as bog haircap moss. And this is a photo of them on the bottom left here. And as these moss grow over time, they form moss peat banks, which are essentially large stacks of moss that accumulate over time. And this is a photo of them on the right. So what makes Antarctic moss peat banks so special is that they are ideal for studying past climate, as the plants in them are well preserved through permafrost freezing. So to focus in on nitrogen in the Antarctic Peninsula, I'm focusing mainly on ammonia volatilization, which occurs when nitrogen is in an organic form known as urea. And this is a process that converts NH4+ into NH3 gas and is transferred into the atmosphere. And it's been found in empirical evidence that soils and vegetation in close proximity to bird and seal rookeries are consistently showing high nitrogen stable isotope values. And 15N, as the heavier isotope of nitrogen, can be used to track movements within the nitrogen cycle. So the figure on the right here just shows a schematic diagram of how ammonia volatilization works. And on the bottom is our delta 15N, how our values were expressed. So our sample is the 15N to 14N ratio, and our standard is air or atmospheric N2. And the equation is multiplied by 1000 to end with the final delta value in per mil units. So the research objectives I had for this project, the first one was to characterize nitrogen abundance in Antarctic land environments by determining nitrogen concentrations in sedimentary profiles. And the second was to investigate the factors that may be affecting the Antarctic terrestrial nitrogen cycle and plant growth. So for our methods, I didn't get to go to Antarctica, but it was still really cool to learn about the field sampling process from my mentor and his team. And these were conducted in 2014 and 2020. And here multiple moss bank cores and living plants were collected across multiple sites. This is a map of all of our 2020 study sites. So our main Cape Tuxin study site is highlighted in the red star. The green circles represent modern plant sampling sites, and our black triangles represent the moss bank coring sites. The brown areas are ice-free areas, and then we also have the land ice in white, and the bluish gray is shelf ice. So this is a really cool video shot by Derek Ford, who was a student under Dave for a while, and he got to go to Antarctica with him. But this is a video of Cape Tuxin, which is one of the largest and greenest ice-free areas on the Western Antarctic Peninsula. It's an ideal study site to track long-term nitrogen cycling due to its elevated and steep location, and this ensures little to no direct input from animals. But it is still located close enough to the coast where nitrogen sources are available and is therefore exposed to nitrogen through processes such as ammonia volatilization. And if you actually see this brown circular area in the middle here, that's actually exactly where the Cape Tuxin moss bank core was taken from. So how our samples were processed, the moss bank samples were first subsampled into 1 centimeter sections. They were baked at 100 degrees Celsius, measured for bulk density, and homogenized. Half of the homogenized samples were baked at 550 degrees Celsius to calculate organic matter content, and the other half was measured and packed into tin capsules and ran through a CosTech elemental analyzer that returned total nitrogen and carbon values. For our plant samples, the Polytricha and strictum leaves were dissected under a stereo microscope, and they were then put through a hot acid treatment to remove inorganic carbon and exogenous debris. They were measured and packed into tin capsules and sent to the Stable Isotope Ratio Facility for Environmental Research at the University of Utah. So for our results, firstly, just going over the modern plant samples, the total nitrogen values ranged from 0.35 to 1.67% nitrogen, and the delta 15N values ranged from negative 0.8 to 14.7 per mil. And these are pretty standard compared to past studies. However, the negative value is a little unexpected, and this reflects a possibly isotopically depleted ammonia source. The sample size was 23, and we found a p-value of 0.534, so no significant relationship was identified between the total nitrogen and delta 15N values. So this is a Cape Tuxin bulk mossbank and foliar core profile. So as you can see from the first graph on the left, it is a highly organic core with almost 100% organic matter. And then we see our total foliar nitrogen and delta 15N trends. And you can kind of see they do follow a pretty similar pattern with a peak in around halfway through, around 115 or so. And we also have the aged up model on the right, and this was calculated through 14C data. And we see that our core is around 3,750 years old, which makes it one of the, it makes it the oldest organic sediment core collected from the Western Antarctic Peninsula, which is also what makes our research really interesting. So for the discussion, a significant relationship between total nitrogen and delta 15N Tux1 foliar leaf values was found with a positive correlation. And essentially periods of higher nitrogen availability are generally corresponding to periods with more 15N enrichment. And once we compared these trends to things like air temperature and carbon accumulation rates, which we have on the two graphs on the right, we compared that to air temperature trends, and it appears that increasing air temperature has increased the process of volatilization, which leads to increase in isotope fractionation, which is leading to delta 15N depletion in Polytrichum leaves at Cape Tuxin. Additionally, carbon accumulation rates are showing that net primary productivity has decreased, which means plants are getting less nitrogen from sources. And this may be attributed to declining animal nutrient excretion due to declining animal populations. So just to conclude everything, so the first point I wanted to make was that nitrogen and delta 15N values have clearly been highly dynamic in the Antarctic Peninsula throughout history. And we see this through especially the depletion of delta 15N values that are correlating with lower total foliar nitrogen. Second point is that depleting delta 15N values are suggesting that volatilization has increased with increasing air temperature over recent decades. The third point is that declining animal populations appear to be reducing nitrogen availability at Cape Tuxin. And lastly, future decreases in marine animal populations will continue to limit terrestrial nitrogen cycle. So I just wanted to acknowledge all of the people involved in this project, NSF and Europe for funding, and then obviously my GES family, as well as everyone from the Biolman lab. So Dave, Lauryn, and Derek, and then our colleagues at the University of Utah Surfer Lab, Suvanker and Ming, and then from the University of Wyoming, Dulcinea, who provided me with a lot of feedback as well. Thank you for listening.
Great job, thank you. Any questions? Okay, must have been a very informative talk. If you have questions.
I can ask a question if you don't mind.
Yes, go ahead.
So Tiana, the nitrogen values are showing potential changes with animal populations. What do you think you would do next if you were going to continue to explore this topic?
Yeah, so I think you and I, Dave, have talked about how running some fecal biomarker data, that would be really cool, and seeing if there is a direct input happening at Cape Tuxin specifically, which would be quite unexpected unless we did see skuas in those videos that may be providing some direct nitrogen input, but that would be really interesting to see.
We have time for one more question if there is one. Okay, thank you. Our next presenter will be introduced by Dr. Megan Donahue from the Hawaii Institute of Marine Biology.
Thanks, Glenn. I'm really pleased to introduce Hanoka today. Hanoka joined our team last spring and worked with us throughout our field sampling season last summer, sampling sediment and crabs and fish in Pu'uloa as part of our Sea Grant project. And she worked really closely with our whole team, including postdoc Dr. Eileen Nally and chemistry graduate student and recent GES grad, Honor Booth. And so we really enjoyed having her as part of our team. And she took on a piece of this larger project to work on, and I'm excited to have her share this work with you today.
Thank you, Megan, for that introduction. And hello, everyone. I'm Hanoka. I will be presenting my thesis on sediment characteristics and contaminant concentrations on Oʻahu and Kauai. Starting off with the introduction of land-based pollution. Land-based pollution describes contaminants that originate from historic or ongoing human activities that occur on land and are transported through watersheds into the nearshore coastal environment. And while there are many forms of land-based pollutant, this project focuses on heavy metals. So heavy metals naturally occur in the Earth's crust, but elevated concentrations are a concern. And these concentrations are typically the result of human activities such as mining, residential or agricultural uses, or industrial processes. And these move through the watershed due to runoff from roads, leaching from landfills, or improper disposal. And once they enter the aquatic environment, these metals may bioaccumulate, which is the pollutants being taken up by organisms and accumulating in their tissue. And these pollutants may also biomagnify, which is when the pollutants travel up the food chain through consumers, resulting in higher tissue concentrations at higher trophic levels. And because humans tend to eat higher trophic level organisms, say predatory fishes like snapper or tuna, humans can be exposed to these higher levels of contamination. So why do we need to understand sediments when we are thinking about pollutants entering food webs? So sediments are a key to the transport and fate of marine pollutants and can help us understand their spatial distribution. Heavy metals are poorly soluble in water, but they can bind to sediment and therefore be transported by sediment into these coastal zones. And the two sediment characteristics that are fundamental to this transport process, which we analyzed in this project, are percent organic matter and grain size distribution. So a little bit about these two sediment characteristics. Grain size distribution is a key characteristic of sediments, and it describes the proportion of sediments in different particle size categories. So smaller grain sizes result in greater surface area for metals to attach, and these small grain sizes are also more easily resuspended and transported through the watershed into the coastal zone. For percent organic matter, we know that sediment is made up of both organic and inorganic matter. Organic matter being the breakdown of plants and other organisms, and can be a significant proportion of the fine sediments. And some metals are more likely to bind to organic matter and therefore can be carried through the watershed. So my work is a part of a broader Sea Grant funded project on land-based pollutants in Hawaiian coral reef ecosystems. And the objectives of the project are to determine the concentration of specific land-based pollutants in at-risk locations on Oʻahu and Kauai, to identify relationships between contaminated coastal sediments and tissue concentrations in fishes and other marine organisms, and to provide key data for evaluating risk to human consumers of coastal fish. And to support this ongoing work, the objectives of my thesis are to analyze the percent organic matter, or POM, of the sediment sampled from these at-risk sites, to understand the relationship between POM and grain size distribution in these sediments, and to examine how these sediment characteristics are related to heavy metal concentrations across the sites. So the sediment samples were collected from 22 locations along the coast of Oʻahu and Kauai, Hawaii. And the first set of samples were collected from at-risk locations around the two islands, focused on areas adjacent to industrial land uses, landfills, or possible agriculture runoff locations. And this round of samples took 3 replicates per location. The second round took a closer look at Mauna Bay with an additional 8 sites in a more targeted effort to look into where runoff occurs. And this round had multi-increments with 3 replicates per location. Jumping into the methods of the sample processing, the first step was to place the samples into the drying oven to evaporate the water out of the samples. Then the samples were weighed. Next, the samples were placed into the muffle furnace to burn off the organic matter from the samples at 500 degrees Celsius for 4 hours. The samples were weighed again, and the differences between the weights of these two were used to calculate the POM of each sample. And as for the grain size analysis, that was done prior to my project. So the results and discussion, first off, we have the mean percent organic matter at each site. The sites are grouped by geographic region. We have here the Mauna Bay locations in blue, the rest of Oʻahu sites in orange, Haina Kauai in green, and the rest of Kauai in red. And across all these samples, the POM ranged from 1.4% to 12.19%, with an overall average of 2.75%. And the location with the highest mean POM was Kāneʻohe Bay, as we can see here, followed by Hanapepe Harbor and Anini. And the location with the lowest mean POM was Haleiwa Harbor. Next, we have the grain size distribution by location, starting from the smaller grain size classes in the darker shades of blue. We have Kāneʻohe Bay with the highest percentages of sediment in the 2 smallest grain size classes. And Kāneʻohe Bay also actually had the highest percentage in the largest size class as well. Next, we have Nāwiliwili with the highest percentage in fine grained sediment. Kekaha and Waimanalo Gulch with the highest percentage in the sand size fractions we see here. And Anini, as well as Wailupe, with the highest percentage in the very coarse sand grain and the granule size classes. Having characterized the sediment at each site, we can now look at the concentrations of the metals. We actually had these sediment samples tested for several metals, but for the purposes of this presentation, I focused on arsenic because it had the strongest relationship to the sediment characteristics. So the threshold effect level, or TEL, and probable effect level, or PEL, are located with these red lines we see here. The TEL indicates the concentration of arsenic in marine sediment at which negative impacts on marine life are possible. And PEL indicates the concentration at which negative impacts of marine life are likely to occur. So the location with the highest mean concentration of arsenic was at Mānoa Bay Beach Park, or MBBP, followed by Hanapipa Harbor and Kāneʻohe Bay. MBBP was a part of the first round of sampling, and the elevated concentrations here, as well as the community interest, is what motivated the follow-up sampling in Mānoa Bay. The high value and high variability at this site was due to a single sample out of the 3 samples with a concentration of 181 milligrams per kilogram, which greatly exceeds the PEL. And this also illustrates the patchy nature of contaminants, because the other 2 samples at this location had a concentration of about 5.5, which falls below the TEL. And as for Hanapipa Harbor and Kāneʻohe Bay, these locations also had a concentration that exceeded the TEL, but for these samples, the variability was much less as compared to MBBP. And all these 3 locations with high concentrations of arsenic were located near harbors, which may be receiving contaminants via runoff and leaching. And our study is not designed to identify the sources of arsenic, but we know that arsenic is used in copper chromatid arsenate, which is a wood preservative that was widely used prior to being banned for residential use, but it is still legal for industrial use. And MBBP as well as Kāneʻohe Bay are residential zones, but the arsenic that we are seeing there may be due to the remains of the old TCA in the area, while Hanapipa Harbor is zoned as industrial. And arsenic is also historically known to be used in agricultural pesticides, which could be a possible, another possible source. And in conclusion, we found that locations with finer sediments also had higher POM, as found in previous studies. As for the heavy metals, arsenic was positively related to the finest grain size category. We also found that particulate organic matter was not a significant predictor of metal concentrations. And we also tested other factors such as region and land use, which also influenced the 3 variables. So for future research, my work will be incorporated into the broader analysis of the tissue concentrations of metals in the fishes at these locations. And the ultimate goal of the bigger project is to provide useful information for resource managers and community organizations that will hopefully aid in future management plans. I would like to acknowledge Eileen, Nayli, Honor Booth, and Megan J. Donahue for all of their guidance and support throughout this entire project. And thank you for listening.
Great job. Any questions?
Yes, I'd like to ask a question. Can you hear me?
Yes.
Okay, first of all, very good presentation, Hanukkah.
Thank
you. Thank you. My question relates to that great variability when you looked at MBBP and you had a high of 41 and a low of 5. Well, first of all, how do you explain that variability, and what did you do about it besides retesting or something?
We looked into Mauna Bay Beach Park, and we saw that the samples, just the location itself has high sedimentation generally. So that's why we were more interested in looking into this location. So Mauna Bay Beach Park, we can see here, is spread out from Kahala all the way to Hawaii Harbor. So we looked at the runoff areas of this location, and yeah, we just found that in general, this area has high sedimentation, which could be the reasoning to the variability of the sites. Did that answer your question?
Yes, except that I wonder, so when you make a conclusion with that kind of variability, what does that mean? That it just kind of depends on the day that you do it, or it depends on exactly where you got the sample or something?
Right. So from my understanding, the high sedimentation in a certain, like in a location, means that it could just have high variability of the concentration, like the metal distribution is just more variable.
I see. And it will be that way whether it's tomorrow, the next day, or next week.
Correct.
I see. Okay. Thank you.
Okay, nice job. Thank you.
Thank you.
To introduce our final speaker, the presenter for the morning session, we have Dr. Prim Das from Urban and Regional Planning.
Aloha. I'm here to introduce Seraphina King. Seraphina is interested in studying environmental sustainability, and she'd like to explore this topic from an urban planning perspective. In fall 2020, she received a Europe Award to study urban agriculture, which culminated in her thesis, and it's been delightful to see her project take shape. This is the first time Seraphina's attempted a qualitative study, so I hope you'll appreciate all the hard work she's put into learning a research method that's different from the lab experiments she's accustomed to in her field. For this study, the city was her lab. I'd now like to invite Seraphina to share her research.
Much for the introduction. So, hi everyone, my name is Seraphina King, and today I'll be presenting my thesis titled Bringing Food to the Table, Exploring the Potential of Urban Agriculture in Honolulu. So just to start off, this is an outline for the following presentation. I'll first cover some of the background information and then go on to the methods as well as the literature review that I did and the findings from my literature review as well as the different interviews. So to start off with, this is a diagram that shows the different components of food systems and the different ways that they interact. So a food system can also be defined as the sum of actors and interactions along a food chain. Climate change can have significant impact on food systems. It can affect the food availability, access to food, how food is able to be utilized, as well as the stability of a food system. It is also impacting where certain crops can be grown. The SDGs, or Sustainable Development Goals, were created by the UN to help increase sustainability globally. One of the goals, all of the goals can be related to food systems. However, the ones that aren't covered with a transparent cube are the ones that most relate to food systems. So the main research question that I focused on during my research is how viable is urban agriculture in Honolulu? While food security is a critical issue and the COVID-19 pandemic has exposed the weaknesses of food supply chains, my study is limited to how urban agriculture can support existing food supply chains by contributing to localized food systems. So to start off with, I first did a literature review of urban agriculture, food systems, zoning, and all of these details that are incorporated within urban agriculture. I then did precedent studies on 3 cities, which were Singapore, Vancouver, and Portland, to get a better understanding. I then sought IRB approval in order to interview key informants from different sectors, including public, private, and nonprofit. The questions for the interviews were drawn up from the literature review and from the precedent studies. So the pink arrows seen on the screen are the step-by-step process, and the tan arrows show how the interview questions were influenced. So why is the study important? Honolulu is not food secure. One of the main reasons for that being that it relies on imports for 85 to 90% of its food. This means that at any given time, there's only enough food in reserves for 5 to 7 days, which is less than the 14-day recommendation. Some of the main issues that the food system here in Hawaii has the first of all is vulnerability. So 48% of families here are food insecure. Native Hawaiian families are also 3 times more likely to face health inequalities from issues relating to food. And the self-sufficiency ratio is 15.7%, whereas the food import dependency ratio is 102.5%. And one of the things that the COVID pandemic has shown is how quickly transportation can shut down. And with this in mind, as well as the limited amount of food available in reserves, looking into other possibilities for food, especially in emergency situations, is important. The full extent of impacts from climate change are unknown, so it is hard to predict the potential ways it could disrupt transportation routes. Increasing local produce in Honolulu, such as through urban agriculture, can help to increase food security as well. So urban agriculture generally refers to the cultivation, processing, and distribution of agriculture products in urban and suburban settings as defined by the US Department of Agriculture. In this project, I defined urban agriculture as food that is able to be grown in an urban landscape with a focus on produce grown by corporations and within community gardens. So the 2 images on the screen show different examples of urban agriculture. So the image on the left is an example of vertical farming using hydroponics. So this is a water-based nutrient uptake method. And on the right, it is an image of a community garden that is located on a rooftop. There are many different types of urban agriculture, which have a vast range of scale as seen with the images on the slide. Some of the most common types of urban agriculture include community gardens, vertical farming, rooftop gardens, peri-urban farming, and private gardens. Different projects will have different goals which help inform which type of urban agriculture to use. In this project, I focused on community gardens, vertical farming, and peri-urban farming. So this table shows the different opportunities and constraints that are related to urban agriculture, and these were found through the literature review that I did at the beginning of this process. So the logos on the screen are different projects that are taking place in Hawaii and in Honolulu. Some of them include the Ulupono Initiative, which has many different projects that are currently taking place relating to increasing local food. The Aloha Plus Challenge has designated one of its goals to local food, and this is production as well as consumption and distribution. Local Foods, Local Places Action Plan is a program that works with different communities to implement different strategies that the community believes will be beneficial to increase local food. And finally, there's the Kāneʻohe Special Design District, which is an area in urban Honolulu that uses a different zoning system. The main focus for this district was to create more mixed-use zones, which allowed for different projects to come into the area that the standard zoning used in Honolulu wouldn't have allowed. So the map on the screen was created using the data from the City and County of Honolulu's GIS program. It shows the different community gardens designated by a green dot, as well as farmers markets designated by a pink dot in the Honolulu area. The number of farmers markets is substantially larger than the number of community gardens. And with respect to the rest of the island, Honolulu has the highest concentration of both. So looking into the location of these elements is important when ensuring that these communities are not being underserved. So these are the different precedent studies that I did, and the cities chosen have been able to successfully implement urban agriculture projects, and therefore would be interesting to see what worked well in the implementation of urban agriculture. So Singapore was chosen to study as it is also an island with limited space. The government in this case played a big role in this by incentivizing the use of urban spaces for production, as well as promoting the consumption of local produce to residents. Vancouver was also an interesting city to look into, as it is one of the leading cities for urban agriculture globally. One of the main things that helped implement these projects was the creation of a completely different zone designated RA, which stands for residential agriculture. And this allowed for urban agriculture to take place in residentially zoned areas, and also for these produce to be sold for a profit. Portland is also a leading city of urban agriculture. However, I chose to include Portland as well as Vancouver because it is located in the US. So there could be similarities when it comes to regulations. In Portland, one of the main driving factors for the urban agriculture implementation were the number of different grassroots organizations, NGOs, and nonprofit organizations. And so you can see on the side, there are 3 different images from each of the cities. So in order to get the data, I interviewed 10 different experts from private, nonprofit, and public stakeholder groups over Zoom. These interviews were then transcribed and the main codes were found and extracted. From these codes, I found the themes that were seen in the different codes. I then found the frequency of those codes across the different, I then found the frequency of those themes across the different codes within the same stakeholder groups, as well as the codes from all of the different transcripts. So as you can see on the right, there are 2 different examples of what the finished product would look like. So at the very top line, you have the main theme, and the bullet points underneath are the different sub-themes that were found in relation to the main theme. And the numbers next to the main theme are the frequency of each of the main themes found throughout all of the different transcripts. So for challenges of implementing urban agriculture, that theme was seen 163 times throughout all the different transcripts of the 10 different interviews. So these bubbles represent the 5 top main themes that were found in the transcripts. So they roughly resemble the frequency. So the highest frequency is challenges of implementing urban agriculture. And some of the sub-themes, as seen in the previous slide, include the limited availability of land, the cost of implementing urban agriculture projects, as well as zoning regulations. So, and then after this, we had encouraging urban agriculture, which looks into the different ways to help increase urban agriculture projects. And this included grants and funding, incentives and subsidies, as well as promoting urban and local agriculture and updating restrictive regulations. After this, we had benefits to urban agriculture, which included environmental sustainability, increasing resiliency, and strengthening the local economy. We then had aspects that can be integrated into different urban agriculture projects. And one of the main ones that came up was technology, as well as traditional food systems in agriculture, which is really important, especially here, due to this cultural significance of agriculture and traditional agricultural techniques. And finally, we had regulations for urban agriculture implementation, which also had to do with land availability and zoning. So these are some of the quotes that I found that were gathered from the different interviews. They touch on some of the sub-themes and themes that were found and mentioned. So the first one relates to the benefits of urban agriculture. The second one relates to zoning and zoning regulations and how that can impact implementation. And the last one talks about purely just implementation and how that can be improved. So throughout this project, I've come to the conclusion that urban agriculture could be a possibility. However, in order for it to be effective and for different projects to actually be implemented, current policies such as the land use ordinance need to be updated to allow for urban agriculture projects. Looking at the Kāneʻohe special zoning area can help shed light on what is possible through changing the zoning ordinance. Increasing the amount of funding available would also be beneficial for projects to start up. And one of the main challenges to finding available land for urban agriculture is the competition for land. there's a big push to use land that is available for housing projects as well as other development projects. And while urban agriculture can seem as a great way to increase food security in Honolulu, there are other ways that this can also have the same outcome. One of the main ways that was brought up during the interviews was to increase the diversification of imports and the sources of these imports. So firstly, I'd like to give a big thanks to my thesis advisor, Dr. Priyam Das, for all her help and contribution. This project would not have been possible without her. I'd also like to thank everyone in the SOAS department for all the guidance and the knowledge shared over the years. And finally, I'd like to thank Europe for funding part of the project and seeing the potential it could have. Thank you.
Are there any questions?
Yeah, I have one. Can I be heard?
Yes, go ahead.
Okay. First, I want to say congratulations, Seraphina. You did a very nice presentation. I enjoyed it. And I have to say that I was shocked for the very first part of your presentation to find out, I mean, I had no idea that 85 to 90% of the food is imported to Hawaii. I mean, that to me is so, and that 48% of the people are food insecure. That's a real high number. So anyway, it was very informative, and I just wanted to say I enjoyed it.
Yeah.
Would it be possible to hear something more about the Kāʻaʻoko special district? It wasn't clear what that was about.
Yeah, so the Kāʻaʻoko Special Design District, it uses a different zoning ordinance that's actually regulated by the state. So it's the, I think it's the Hawaii Development Authority. I'm not 100% sure about that. They regulate that, and it allows for mixed use zoning. So this means that instead of just the standard residential or industrial or commercial, you can have a multitude of different, types of projects and development coming in. So this allows for more different types of parks, as well as like different types of commercial spaces that aren't quite orthodox and don't quite fit in with kind of a standard city layout. So it allows for more flexibility in the different projects that can come into the area.
Thank you.
Okay, if there are no final questions, please join me in thanking all the speakers. I believe it was a number of very good talks, or they're all very good talks on a wide range of very interesting science being done through the GES program. So, thank you all the speakers. The second session will start at 1:30 this afternoon. It is a different Zoom link. Latina, do you want to copy the Zoom link for this afternoon into the chat just so that everybody has it? But would be in your various emails. Okay, thank you all for coming, and see you this afternoon.